Hello everyone. Last time we reviewed what the DNA molecule looked like and what it is composed of, and we are reminded that it is composed of nucleotides, deoxyribose sugar, phosphate group, and nitrogen base. We also learned that the molecule itself, each side has a 5' and a 3' end, and basically it's anti-parallel. Each side goes opposite. So we're going to continue with this mini lecture by continuing the animation. It's going to complete the DNA replication part, and I'll begin that right now.
The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions.
All right. Helicase enzyme basically opens the DNA molecule at the replication fork. Okay, that's what helicase does. This other protein out here, it just keeps the, see how the DNA is all coiled up here? Kind of keeps it from coiling up. Kind of just keeps it smooth running so the helicase can come in and open up the DNA molecule, breaking those hydrogen bonds at the replication fork. Let's continue.
DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA.
All right, so did everyone catch that? DNA polymerase is what attracts the DNA nucleotides to make the new daughter strand. Okay, you got a parent strand and you're generating a daughter strand.
Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer.
All right, so the RNA polymerase brings in the RNA primer. Okay, the RNA primase brings in the RNA primer. That is needed to attract the DNA polymerase, which then, attracts the DNA nucleotides to make the daughter strand. Let's continue.
Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
So, the replication of DNA, when it finishes, we have 2 identical copies of DNA, and 1/2 of this DNA is a parent strand, 1/2 of this is a daughter strand, 1/2 of this is a parent strand, and 1/2 of this is a daughter strand, and this is called semi-conservative replication. This also occurs in the S phase of the cell cycle. Okay, that's when the cell is getting ready to divide. It first needs to make a genetic copy, has to have the all of the DNA copied so that at the end of mitosis it will be able to provide enough genetic information to each of the daughter cells. Okay, so this is DNA replication. It occurs in the synthesis phase, the S phase of the cell cycle. So that's how we can tie this whole, you learned way back in a few chapters, way back a few chapters ago, you learned that the cell cycle had various stages, and one of those stages was the synthesis phase. Well, this is how DNA replicates. So hopefully this helped you a little bit, and don't forget that your publisher has all of these animations out in the online learning center. So you just click to the appropriate chapter and then select the animation you'd like to see. Have a good day.